Hello everyone, welcome to Great Online Training. Today's video is about clinical data management training. Let me introduce myself. I'm Anamika. I have 7 plus years of experience in clinical data management, currently working as Principal Clinical Data Manager. I've worked with different clients and well versed with Oracle Clinical, Medidata, Rave, and Inform clinical databases. I'm with Great Online Training from last few years as a trainer. If you have any queries related to this training program, please contact below numbers displayed in this slide. Else, please leave a comment below in the comment section so that I'll get back to you as soon as possible. Let's begin this video. After the end of the video, you'll be able to understand what is clinical trial, what is clinical data management, about our training program, scope of CDM, data management activities, question and answers. So let's start this video. Glossary of clinical trials. As this video is about people who want to begin career in CDM, so some of you might not aware of the terms which are used in clinical research industry. Let me take you through those terms. Starting with contract research organization. What is CRO? Let me give you a brief idea about what a CRO does and how it functions. CRO is basically a company that helps conducting research for a sponsor company. What you must be thinking is, why does a sponsor company need a CRO to conduct its research? Answer to this question is, CRO specializes in conducting research compared to pharmaceutical companies. It also helps the sponsor company by saving cost and helping reducing the time period required to conduct the clinical trial. I hope now you understand the definition of contract research organization. What is informed consent? It's nothing but verification of a person's willingness to participate in a research. Prior to enrollment into a clinical trial, researchers inform participants about all relevant details. Participants will be given informed consent document that details all the important study information, for example, purpose of trial, duration of the trial, risks involved in the trial, potential benefits, and then they will be asked to sign this document. If they agree to participate in the trial, they are going to sign the document. Informed consent document, it's not a contract. So participation in the clinical trial is voluntary and the patient may withdraw from the trial at any time. Institutional Review Board IRB. What is IRB? It's an administrative body established to protect the rights and welfare of human. It ensures that a clinical trial is ethical and rights of study participants are protected. They often conduct some form of risk-benefit analysis. Now, what is risk-benefit analysis? The analysis which focuses not just on the risks of the activity but also on the benefits of the activity to determine whether research should be completed or not. Moving to next slide. Who is a subject? We all know what does a patient is, but in terms of clinical research terminology, an individual seeking medical care, we also known as subject. Next, what is protocol? A protocol is the study plan on which a clinical trial is based. It describes what type of people may participate in the trial, the schedule of tests to be conducted in the trial, procedures to be undergone, example, medications, dosages, and length of the study. Everything will be given in detail in this protocol. Next, who is a sponsor? We have already discussed in the earlier slide. It can be a person or organization who manages and finances a clinical trial. Source data. Records on which clinical observations are first recorded. For example, it can be a signed informed consent forms, any hospital records, laboratory reports. What is case report form? There are two types of case report forms, paper and electronic. Sponsor of the clinical trial uses CRF to collect data from each patient. Adverse event. What is adverse event? Adverse events are also known as side effects, and these side effects could be due to administering the experimental drug or it may not be. For example, vomiting or headache. Some patients are allergic to drugs. Then they might experience side effects or also known as adverse events. Who is a clinical research associate? The person who's employed by the study sponsor to monitor a clinical trial. The CRA is responsible for ensuring all clinical studies that are conducted according to the study protocol and ICH guidelines. Who is clinical investigator? A medical researcher who is in charge of carrying out entire clinical trial. And who is a clinical research coordinator, CRC? The person who is responsible for conducting clinical trials using good clinical practices. I hope I have covered some of the important terminologies we use in clinical research. Moving to next slide. What is clinical trial? You all must be heard about the term clinical trial, but what exactly is clinical trial? Clinical trials are an important step in discovering new treatments as well as new or better ways to prevent, diagnose, or treat diseases. It's nothing but a scientific study designed to examine whether potential new medical treatments are safe and effective in humans. Let's discuss in detail in next slide why we need clinical trials. So new tests and treatments will not be released into the market as soon as they make. They need to be studied. By performing clinical trials, sponsor will come to know how safe and helpful are in humans. When found to be safe and helpful, they may become tomorrow's standard care of treatment. Clinical trials need to be approved before they can start. And they need sponsor since they can be done without money or other support. Sponsors often are government agencies, pharmaceutical or biotechnology companies. Clinical trials also need approval from IRB, Institutional Review Board. IRB makes sure the right and welfare of patients are protected. Once sponsor gets approval from IRB, IRB reviews its progress at least every year. Clinical trials have risks too. For example, some treatments may have side effects, some may not work as good as or better than the one now in use. So now you might having a question like clinical trials, why they are to be performed in humans only, not in animals. The answer is correlation of effects in animals and humans not always possible, and because of the species difference, some effects in only in humans. So man is final experimental animal to be tested. I hope now you are aware of the clinical trial definition and the process. Moving to next slide, clinical trial process. It begins with preclinical testing. What is preclinical testing? Preclinical testing is conducted in animals. It's a stage of research that begins before clinical trials can begin, performed on animals, and these are required before clinical trials in humans can be started. While preclinical research usually provides some general information about dosing, so sponsors or drug developers must submit IND, investigational new drug application, to health authorities such as FDA, Food Drug Administration US FDA, before beginning clinical research. Trials can start once approval is granted. Now we'll discuss about investigational new drug application. It's nothing but a petition through which a drug sponsor requests the FDA to allow human testing of a new drug. Once IND is filed and approval is granted, then sponsor starts phase 1, phase 2, phase 3, and the sixth phase is new drug application. What is new drug application? An application submitted by a sponsor to the FDA for approval to market a new drug for human use. So once sponsor files NDA and gets approval, then commences phase 4. So all these phases, let's see in detail in the next slide. So we have 4 phases. In phase 1, it generally conducted in several months. So 20 to 80 people around are going to be participated in the phase 1 trial. And this phase 1 trial tells about side effects, finding out about side effects and what happens to the treatment in the body. Phase 2 trials we generally call as safety and dosing phase. Several months to 2 years is the period which it occurs. 100 to 300 participants will be there in phase 2 trial. So it says about more about side effects, finding out more about side effects and effectiveness, and looking at how well the treatment is working. And phase 3 trials, in which safety and efficacy will be assessed, and the ratio of the participants 300 to 3000 participants. It compares the new treatment to the standard treatment. Then phase 4. Phase 4, it tells about more about long-term benefits and side effects. So it's a phase finding out more long-term benefits and side effects. So with this, we have completed all the 4 phases in clinical trials. Let's move to next slide. What is clinical data management? Clinical data management is the process of collection, integration, and validation of clinical trial data. Clinical data management is the process of ensuring that data collected during a clinical trial is accurate, complete, logical, and consistent. CDM plays an essential role in the data collection phase of clinical research. The process of collecting and managing research data is done in accordance with regulatory standards to obtain quality information that is complete and error-free. The goal is to gather as much of data as possible for analysis for the regulatory submission. We'll discuss in detail in upcoming slides. Next, data capture. What is data capture? The clinical trial data is collected in CRFs, case report forms, that should be transcribed into clinical data management systems for validation and analysis. So data capture is of 3 types: paper data capture, electronic data capture, and remote data capture. So each data capture we are going to discuss in upcoming slide in detail. So here we have differences between data capture tools, three different data capture tools: paper, electronic, and RDC. In paper data capture, data is entered on paper case report form. Data entry associate will enter the data into the clinical database. What is data entry? Data entry is a process of entering or transferring data from case report form to clinical data management system CDMS such as Oracle Clinical, Inform, or Medidata Ray. So single data entry and double data entry will be performed in case of paper data capture. And disadvantage of this paper data capture is no real-time access to the data and it takes more time. So generally sponsor prefers EDC, electronic data capture tools. In electronic data capture, data is captured in electronic case report form. Then investigator enters the data into the database, where in case of paper data capture, CRFs will be sent to data manager and data entry associate will enter. But in case of electronic data capture, investigator enters the CRF data directly into the database. So we have real-time access to the data. Coming to remote data capture. In remote data capture, also data is captured in electronic case report form. Then system allows research staff or site staff to enter data directly into the system. This can be useful in case of multi-center studies where there can be patients enrolling from multiple sites. So study is being conducted with many institutions participating or multi-centered trial. In this case of setting, remote data capture is useful. It's a web-based, so no real-time access to the data like electronic data capture. I hope now you understood the difference between paper, electronic, and remote data capture. What clinical data manager do? What clinical data manager do? One sponsor finalizes the trial protocol. Investigator selection is done by sponsor. Approval process from IRB and regulatory authorities. Patient recruitment will be commenced. Once subject starts enrolling, clinical trial data gets collected into the case report form, and case report forms will be sent to data managers in case of paper studies. In case of electronic data capture, as we have discussed in previous slide, investigator is going to enter the CRF data directly into the database. Clinical data manager is going to perform data entry and validation. DB load and DB lock performed by CDM people. Data analysis reporting is the next step after DB load and DB lock. Extraction of data which results in meeting. Final report submission to FDA. So let's discuss in detail what exactly clinical data manager is going to perform during the clinical trial process. Clinical data management overview. Once subject starts enrolling into the trial, then investigator is going to administer them. Once subject starts enrolling into the clinical trial, investigator is going to administer the treatment and collect the patient data. So the data is collected is recorded on the CRS and the lab samples are sent to the laboratory for assessment. So here you can see CRF and DCF. DCF is nothing but data clarification form. In case of paper studies, data clarification forms are nothing but the clarifications which the clinical data manager require from the site. So they are going to write their queries in data clarification forms and send it to site. And again, site writing their clarifications, again they are going to send it back to data managers. Here in case of electronic data capture, queries will be generated. So we'll discuss in detail in upcoming slides. So data collected is recorded in the CRFs and lab samples are sent to the laboratory for assessment. Data recorded on the CRF by investigator is verified by the site monitor against the source documents. Both data from the CRF and laboratory are then sent to data manager who runs validation checks on the data and performs data cleaning. So what is validation checks? We are going to discuss in the upcoming slides. As a result of data cleaning activities, data clarification forms are generated as we have already discussed. So are generated which are sent to the investigator for clarification. The database is updated based upon the resolutions received. The clean data is then sent to the statistician who then analyzes the data. Clinical study reports are created by medical writer based upon the statistical analysis results which is submitted to the regulatory agencies for approval. Clinical trials often involve patients with specific health conditions who then benefit from receiving unavailable treatments. So, while initiating the clinical trial, sponsor or investigator first identifies the medication to be tested, and then sponsor decides what to compare the new molecule with and what kind of patients might benefit from the medication or device. So, this is the overview of clinical data management. Moving to next slide, objectives of CDM. We have already discussed about different types of data collection through various data capture systems like paper, electronic, and remote data capture. And what is data integration? So, integration of clinical data means to ensure that data incorporated into the CDMS, the clinical data management systems, are correct, consistent, and it's an exact replica of the data received on the case report forms or paper forms. So, next step in the data management process is the validation of data entered into the system. Validation is done through user acceptance testing methods, what we call as UAT. We'll discuss in detail. While the clinical data is validated through electronic programs written by the programmer. So each step we are going to discuss in detail in the upcoming slides. Moving to next slide. Interdependent groups in CDM. So we'll discuss the various cross-functional groups we work with in CDM day-to-day activities. Data management activity begins with the final approval of protocol, as we have already discussed. All data management activities are performed closely with the programmer and biostats team. Programming team helps to program the validation and derivation checks. What are validation and derivation checks? We are going to discuss in the upcoming slide. And these validation and derivation checks are required to identify the discrepancies in the database. So we need programmers' help for that. When these validation checks are executed, discrepancies are generated in the clinical data management system. So discrepancies will be actioned by the data managers. So clean data is provided to the biostats team, and this step is the final step when entire clinical data is clean and we are ensuring clean data from our end. We're going to provide the clean data to the biostats team at the end of the study, and they perform data analysis. The results of data analysis are used for generation of clinical study reports, and those reports are going to be submitted to the regulatory authorities. So let's move to next slide. So CDM activities phase wise, we have three phases: startup phase, conduct phase, and closeout phase. We'll discuss each phase separately in upcoming slides. Starting with startup phase. Once we receive protocol from the sponsor, we'll initiate the CRF designing. So we have already discussed what is CRF. Again, repeating. CRF, it helps in collecting data, patient data from the investigational sites. So CRF is a data reporting document used in a clinical study. The main purpose is to collect the study data in a specific format that allows for efficient data analysis, which is our ultimate goal. All data must be collected on the CRF if specified in the protocol, which is not mentioned in the protocol, it shouldn't be included in the CRF. So we have to follow some guidelines while designing the CRF, and important guidelines such as collecting the data with all users in mind, because there will be multiple persons accessing the CRFs, collecting data required by the regulatory agencies which requires for the analysis purpose. We have to avoid duplication, and CRF should be clear and concise. You should also capture all information required by the protocol, and it should meet the needs of different clients and end users. CRF needs to be finalized and available before an investigator starts enrolling patients into the study, because as soon as they enroll the patients into the study, they'll start capturing the patient data in the CRFs. So it should be finalized before patient enrollment. Once the draft CRF is ready, it needs to be sent for the client approval. If there are any findings or there need to be any modification, sponsor will suggest us to do those changes. If there are no findings or no comments received from the sponsor, then we can sponsor finalizes the CRF and we can go ahead for the next step, that is database designing. So what is this database designing? Creation of database using clinical data management systems like Oracle Clinical, Medidata, Rave, Inform is called database designing. So who does this? Database programmers will design the database. Database programmer creates the exact replica of CRF pages in the database so that data entry person can enter the data recorded on the CRF. So pages that are entered into the database, it should be according to the data entry guidelines. Once database is designed, database programmer designs the database. As a data manager, we have to test the database. Database testing needs to be performed prior to activation of database. In case of any findings, database designer will fix the issue. Database can be activated in case of any findings. He is going to fix the issue. If there is no finding, database can be activated. Once all the findings have been resolved, then database can be activated. What is the next step? Validation and derivation procedures or programming procedures. The automated edit checks, we also call as automated edit checks. Automated edit checks, these are required to flag errors or discrepancy in the data present on the CRF or in the database. So these we call as validation procedures or edit checks, and these will flag the errors. So once edit check programmer designs the program, like programming the validation procedures, it's an important activity to be completed during study setup phase. And the process for this validation and derivation procedures, edit check programmer writes validation procedures. And the logics which were given by data managers, edit check programmer writes the validation procedures. So, data reviewer enters clean and unclean data to test whether these checks are firing fine or not. What we call as user acceptance testing. User acceptance testing UAT has to be performed for all the checks, and this is the final step before releasing the application or database to sites for capturing the clinical trial data. So UAT is the last step in the setup process. So UAT is performed while entering clean and dirty data. Clean data, the objective is to we have to get zero errors, and unclean data to flag the discrepancies, and just it's the purpose of testing the check whether it's working fine or not as what we are expecting. According to that, we need to check before database gets activated. So once all these checks will be fine, when all these checks are working fine, then database gets activated and it's ready to accept the production data. So this is the entire process of study startup. Moving to next slide. Different roles and responsibilities in study setup. CRF designers, who designs the CRF as per the protocol, and DB designers, who design the database and activates. Programmers, as we have discussed, who programs the validation and derivation procedures and activates the checks. Data managers. Data managers are required in each and every step in the setup process. While designing the CRFs, before the finalization of the CRF, data managers has to review the CRF, and before activating the database, DB has to be tested by data managers, and the validation and derivation procedures, the logics has to be given by program, like logics has to be given by the clinical data manager before activating the checks, and also performs UAT of the checks before activation of the database to make sure all the edit checks are working fine or not. Next slide. Study conduct process. Once DB is activated, data entry is done. Again, in case of paper studies, CRFs will be sent to the data managers and data entry person has to enter the data in the database. In case of EDC studies, investigational sites enter subject data directly into the database. And here you can see CRF and external data. Now you may have a question like, what is external data? External data is nothing but non-CRF data like third-party vendor data. In trial protocol, if we are sending any samples to the lab or ECG data, IVRS data, those data we call it as external data or third-party data. So then once data is entered, discrepancy management would be performed by the data manager. So what is discrepancy management? It is the process of identifying and managing potential problems with data collected during a study. For example, I can say like empty fields, unrelated item or incorrect ranges, lab ranges, or dates not in logical sequence, any missing visits or pages. These all come under discrepancy management. So these are the error data which will be entered in the database. So apart from that, coders will perform coding of the data, the medical terms or any concomitant medication or any drug name should be entered in the database. Coding would be performed by the medical coding would be performed by the coders. and simultaneously safety data reconciliation. What is safety data reconciliation? It's a comparison of clinical database and safety database, because majority of the data includes the safety terms, the adverse event terms, which needs to be reconciled. So we are going to reconcile and perform this activity, safety data reconciliation. So while we are performing the discrepancy management, coding and safety data recon, we can identify number of issues out of these activities. So queries would be generated while performing these activities, and the queries would be generated in two ways: through automated checks, what we have seen in setup process, through validation and derivation procedures, which flags the errors whenever site enters data. These procedures in the back end of the database, they'll be running and they'll be generating the queries in the database. In case of manual check or QC, data manager has to do the manual review, because for particular data only we are going to implement the edit checks. For rest of the data, we need to do the manual review. In case of query generation, where we have automated checks in place, queries would be generated and those would be sent to site for clarification. Site comes back with some resolution, or if any modification is required, site is going to update the data. As data managers, we do not have an access to do any modifications in the CRF. It's just our job to review and clean the data. We do not have any access to alter or do any modifications to the CRF. We have only read-only access and have access to raise the queries to the site. When site comes back with some resolution, if there is really a need of changing the data, site may change the data, else they can confirm the data as is. So query generation and query management is done in this way. So once resolution and update of database is done, then we can close the issue and make sure data is clean. So this is an overview of conduct process. So I hope now you're aware of study setup and conduct process. Now moving to last phase, that is study closeout. I hope now you're aware of study setup and conduct process. Now moving to next slide, different roles and responsibilities in conduct phase. Data entry or data loaders, we have already discussed. Person who manually enters the data from case report form to the clinical data management systems and who enters the external data, examples lab or subject diaries. And data managers, so we are going to identify and resolve discrepancies in this conduct phase. Majority of the data gets cleaned during conduct phase. We are going to issue queries and resolve them following with site, and also perform manual review through manual checks. In case of paper studies, CRF tracking is done to resolve the issues. Safety data managers, who performs the comparison of safety database and clinical database. And dictionary coders who code the medical terms collected during the clinical trial. So most of the terms would be captured in the adverse event form and concomitant medication forms. Concomitant medication forms is nothing but the drugs which would be administered during the trial apart from the treatment drug. Next slide. Study closeout process, and this is the last step, last phase in the clinical data management system. So what is lock? When all clinical trial data has been reviewed, queries resolved and issues addressed, then database will be locked. So database cannot be changed in any manner after locking. So when we are approaching the lock phase or the final lock, we have to ensure that we are meeting some of the pre-lock activities. We have to refer the checklist for locking the database, and the checklist says like all data has been entered and we didn't miss any data, and site has entered all the data we have reviewed and cleaned. So all data entered and processed, all the coding of clinical events is complete, and reconciliation between database and SA system or any external data we have to perform frequently, so that there should not be any outstanding issues and each and every should be resolved by this time. So all queries are resolved and database is updated. So QC audit would be performed and issues will be addressed after performing the QC. So issues addressed would be intimated by the sponsor or internal audit team. And again, we have to get the approvals from the investigator. So investigator is going to sign the CRF before locking of the database. So they have to verify the CRF data, whatever the data entered in the database, they have to compare with the source document, what we call as source document verification. So this is the last step. Once we are ensuring that entire data is clean, error free, and nothing is outstanding, if there are any outstanding issues, we have to document it before locking of the database and get approval from the sponsor. So source document verification would be done by the site persons, and final step is investigator approval. Investigator is going to give their signatures in the CRFs, investigator signatures. Once investigator signs the CRF, then we can go ahead and lock, freeze and lock the CRFs, so that site cannot modify and they lose the access of modifying the data in the CRFs. So this is the process of study closeout. In this study closeout process, whatever activities we are performing in conduct, we are again increasing the frequency and performing here also. As you can see, safety data reconciliation, discrepancy management, coding terms, query generation, resolution. We are following the same steps just to ensure that we are not missing out any data and our data is clean and error free. So this is the study closeout process review. I hope now you're aware of study setup, closeout, and conduct. Moving to next slide. Different clinical data management systems used in current market: Oracle Clinical, Medidata Rave, Inform, Data Labs. These are few CDMS tools which are used frequently and in current in CDM industry. Once you complete this training, you are able to get jobs in these companies which were listed in this slide. Moving to next slide. These are the salaries, approximate salaries for data managers according to the experience or the level of data managers, like data manager and senior data manager. If you have any questions, please comment below in the below comment section. So regarding our training program, if you have any questions or regarding this presentation, please get back to me provided in the numbers in displayed in this slide. Thank you so much.